PGS is pre-implantation genetic screening, and nowadays that's more commonly called PGT-A. So that's pre-implantation genetic testing for aneuploidies. So this means you're testing each of the embryos that you make to ensure that the chromosomes for that embryo are correct both in number and appearance. So currently, embryologists use two moments in embryo life when the chromosomal load can be assessed, and that can be done on day 3 or day 5 of embryo life. On day 3, an embryo is in 8 cells. So what they do, the embryologists do, is use a laser to make a small hole in the embryo's shell to extract just one of those cells. So leave 7 behind and take away 1. This biopsy doesn't seem to hurt the embryo, it doesn't seem to stop it from evolving, and the chromosomes in the cell are removed and then analysed, and we get the result within about 48 hours. And if we find that the embryo is genetically normal and it's continued to evolve in the lab, then we can transfer it then, and it's a fresh transfer into the patient's uterus. The embryo biopsy on day 5 takes place when the embryo is at the blastocyst stage. So the embryologist in this case will have weakened the wall a little bit on day 3, then made not a direct hole, just weakened the wall of the shell on day 3. And as the embryo has evolved to day 5, it will push through that weakness, and some of the outer cells of the embryo will come out. So that's the outer part of the embryo at that stage is called the trophoectoderm. And on day 5, the embryologist can then take 4 or 5 cells of the bit that's sticking out without actually touching the inner cell mass, which is the bit inside the embryo, which is later going to become the fetus. And so then they freeze the embryo, and then we can analyze the chromosomal load from those cells that have been removed. And it usually takes up to about 2 weeks to get that result. And then we can do a frozen embryo transfer at a later date if the embryo is found to be genetically normal. So what are the pros and cons of these two different approaches? So the day 3 biopsy gives us a very quick result, so 48 hours, and allows us to do a fresh embryo transfer, which in egg donation still gives us a very slightly higher chance of success. That may not be true in IVF. Egg donation is slightly different. However, whilst we know that the embryos that have been biopsied on day 3 that are normal are definitely normal, they're shown to be normal on day 5 as well, there's some evidence that the embryos biopsied on day 3 and diagnosed as abnormal some of them, actually self-correct before day 5. So we could potentially be discarding some viable embryos, a small proportion of viable embryos. So the day 5 biopsy gives us a more reliable result because 4 to 5 cells have been taken, but there is still a question actually with day 3 as well as to whether the cells we've taken are representative of the rest of the embryo. And we also aren't quite certain whether an abnormality detected on the outer cells, so the trophoectoderm cells, is really a bad thing, because we know that you can transfer embryos that are known to be mosaic, so there's a part that's normal and a part that's abnormal, that can create healthy live births. So essentially, how is it performed? They do the biopsies and then they analyze it, and that's the process. And it's difficult to know which of the 2 ways of analyzing the embryos are currently better, which one of the 2 is better.